Okay, this podcast is dealing with Lab 2, which was in fact the first lab we did because we did this as part of our biochemistry unit. If you'll recall, biochemistry dealt, of course, with inorganic and organic compounds, the four major groups of our organic compounds being carbohydrates, lipids, proteins, nucleic acids. And this lab focused on protein, one in particular very important group of proteins, which we know as enzymes. We looked at how the rate of an enzyme catalyzed reaction and things that can affect the rate of an enzyme catalyzed reaction, how fast that reaction can occur. The lab setup for this lab, well, let me back up just a bit. The enzyme of interest in this lab basically is picked because it's an easy enzyme to obtain. The enzyme involved is called catalase, and its job is to take hydrogen peroxide, which is highly toxic to cells but is made by cells as part of their regular metabolism, and to break it down into water and oxygen, and not a balanced equation, but that's okay. Now, just an aside, there is an organelle in your cells whose job it is to contain catalase that will break down this nasty toxic hydrogen peroxide into these two harmless substances. That organelle is called a peroxisome. Makes sense, peroxide, peroxisome, and that organelle is simply a sac that contains catalase that will degrade this hydrogen peroxide for us. Now, most all living things, at least a large majority of living things, make catalase because this problem is so universal. And there are many ways that you can measure how fast an enzyme is working. You can measure how much of the reactant gets used up. You can measure how much product gets made. Now, given that hydrogen peroxide and water both look a lot alike, and we're doing this all in a test tube, it's kind of difficult to measure how much of this gets used or how much of that gets made because they're both clear liquids, they're in the same tube, difficult to measure. What is very easy to measure, though, is the gas, the oxygen gas that comes off, because that will rise up out of the liquid in bubble form. Okay, so the oxygen produces bubbles in the mixture as the reaction is happening. The way we chose to do, or I chose to make you do this lab, was we had a reaction vessel. It looked a little different from this, but a reaction chamber where we placed some hydrogen peroxide and some catalase. And as those two things reacted together, bubbles was connected by a hose that went up into the graduated cylinder. Now, the graduated cylinder had been placed in water and pulled out of water, so it was full of water. And as the bubbles went up into the tube, the water was displaced out. And you could easily measure how much gas was being produced by the water being driven out of the tube. And you could just measure where the gas was in the tube. Okay, here you can see that happening here. All right, so we measured the gas in the graduated cylinder. Okay, so that's again pretty much showing us what we did. Now, the concepts of this lab. Okay, you need to know what a substrate is. That's the thing the enzyme acts upon that might fit into the active site of an enzyme. So here's a substrate, and it might fit into the active site, which is right there in this particular model of catalase. Here's a easier to understand model here of an enzyme and a substrate and an active site. You need to know what an enzyme is. That active site structure and all that, you should also remember about enzymes, that they are not broken down by the reaction they catalyze, so they can be reused over and over and over and over again, that the things that they create are called products, that enzymes can work by breaking down something, as is in this case, or by taking two things and putting them together. The enzyme speeds up a reaction by forcing things together or weakening bonds and breaking things apart. Okay? You should know what this term means, denaturation of a protein, what it means for a protein to be denatured, and the things that can denature proteins. For example, extreme temperature, mainly extreme high temperature. Remember that cold slows down chemical reactions because it slows down the amount of collisions, the kinetic energy collisions of molecules between each other, but cold doesn't usually denature the enzyme. Heat denatures enzymes because as the molecules begin to move, bonds get broken within the enzyme. The hydrogen bonds or the other bonds, usually hydrogen, break first, that might be holding this thing in its particular shape. Remember the key analogy that we used. That shape is very, very important to enzyme function. Okay. Other things that could denature proteins are extreme pH, even extreme salinity. And in fact, when we did the lab, we exposed some catalase to high temperatures. We exposed some to acid versus base pH. We changed the concentration of the enzyme. We put more enzyme in the reaction chamber and determined if that made the reaction go faster. We put more substrate in. We did a lot of things to see how it would affect the rate of reaction. The experimental design. Let me get all these up here. Okay. This is what I was just talking about, so I hate to repeat myself, but the rate of reactivity is what we were measuring. This is your dependent variable. We were looking at how various things affected the rate of enzyme activity, which we measured as being the amount of oxygen produced over time. We tried to look at the effects of pH, or the effects of temperature, the effects of enzyme concentration, how much enzyme we had, a lot or a little, how much substrate we had, a lot or a little. These kinds of things, pH, temperature, amount of enzyme, these are things that could all be independent variables in an experiment. These are what we manipulated. These are what we measured. Okay. Typically, what we found was enzyme reaction was affected by pH and temperature and how much substrate was present. The more substrate, the more the enzyme can bump into things, and so it will be faster if there's only a little bit of substrate. The more enzyme present, the more enzyme there is to bump into substrate molecules, so that would increase the reaction rate, and so on. You should understand how what you're reading in this graph right here is evidently an optimum temperature here for this particular enzyme. Normally, we would imagine optimum temperatures would not be this high. We would think they would probably be around body temperature because, or perhaps because that's the temperature that most enzymes are designed to interact with. Let's see. Rate calculation. Please remember how to calculate a rate. It is this formula. We've used it several times over the course of the year. It's slope. Okay, so usually this might be your your last data point and your first data point, and this would be the matching x value here. So last minus first, depending on what you're trying to measure. But obviously, if they want to know what is the rate between 10 and 20 seconds, then you would take these as your data points, or 0 and 20 seconds, then you would take this and this as your data points. So you have to kind of pay attention to what they might be asking for. Okay? And don't forget your units. And please remember how to draw a graph, and y-axis and x-axis need to be scaled properly, to be labeled properly with a label and a unit, a label and a unit, a title. You get points for these things, not to mention a correct plotting of the points. When in doubt, don't draw lines. Certainly don't extend lines out past whatever data points you have. If this is your graph, and you have these data points here, 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 and here, that's your line. Don't go out past that. Don't assume. You might, if it's relevant, show a dashed line that is what you might predict, but don't assume that you need to fill in where there is no data. This is an essay. I don't remember if we actually did this one or not. Effects of pH and temperature were studied. These are the results. How do temperature and pH affect the activity of this enzyme? Discuss the relationship between the structure and function of the enzyme, as well as discussion of how structure and function of enzymes are affected by temperature and pH. This is talking about the importance of a shape of enzyme, its active site, and the effects of denaturing on that, the breaking of bonds that are holding that tertiary or quaternary structure. You might even talk about those kinds of things. And then this is asking you to basically describe a controlled experiment where you could have produced the data that is shown for one of these. So we did this in the lab. And it says, be sure to state the hypothesis that was tested here. So state a hypothesis. There's a point right there. Okay, so that's that. Let's see if there's... Okay, that's it. So end of lab 2. Let me know at review if you've got questions. Thanks for watching.